Hey there. So I wanted to carry on with our lecture on the catabolism of amino acids. And now we've discussed what happens to the nitrogen and removing that nitrogen from the amine group of the amino acids. And we've taken it off. What we're then going to do with that and how that gets integrated into urea for the ureatelic animals such as mammals, and how that gets added to urea and then excreted out of the body as waste product. And I wanted to talk about that process and the urea cycle, which makes up that process. So, building on the concepts that we had before, there are the several key concepts that I presented before. And of those, we'll be looking at in particular concept 2, 3, and 4, which are that there's several key amino acids which are involved in transporting nitrogen around in the body and excess nitrogen. And these are alanine, glutamate, glutamine, and aspartate. Transports nitrogen around the body so that it can be used or excreted. The metabolic pathways are not separated. And that will become evident later, but it's the idea that a lot of these pathways are linked together. For instance, the TCA cycle, lots of things go into the TCA cycle, lots of things come out of the TCA cycle, and that can be really changed in order to suit the needs of the cell, suit the needs of the organism. For instance, what we're going to learn with the urea cycle is that something's going to come out of the TCA cycle, but then get returned back to it. And then later on in the lecture, I'll talk about how different amino acids can be broken down, and the carbon part of the amino acids is broken down and integrated into the TCA cycle. Or for instance, maybe you need to take out certain TCA cycle intermediates in order to make amino acids for certain things. So that's going to be covered in this lecture as well. And the whole point of it is because ammonia is toxic. And so when we metabolize amino acids, like I was saying in the last lecture, the first step is removing the amine group, but we need to do something with that amine group, and it can't just sit around. And if the amine levels increase, the ammonia levels increase, then that's typically toxic to animals and cells. And so that ammonia needs to do something with it. And the best thing to do with it is to turn it into urea and then excrete it. And urea is good for that because it's non-reactive in this context. When levels increase, then yes, it will become toxic, but it's significantly less toxic than ammonia. Also, it's very water soluble, so it's easily excreted. So to get into it, the urea cycle is the way in which ammonia is taken and converted to urea, mainly utilizing the mitochondria of hepatocytes, which are liver cells. As with other cycles, metabolites are produced and they will cycle around, and some things go in and some things come out, such as, for instance, urea comes out, but at the end, we return to the beginning of the cycle. And so it's the same kind of thing. We start with an empty molecule, you add something to that empty molecule, it goes around, and then something comes off that molecule, and it is restored back to the beginning, kind of like I say, restored. You get back at the beginning that beginning metabolite. And like I say, urea enters the bloodstream and then is excreted in the urine. So one of the key things for the urea cycle is that it does require energy. So you are going to require ATP as part of this process, and that will come apparent later. And two nitrogens are going to be lost because the structure of urea is, so it starts with this carbonyl, and you have these two amines, sorry, NH2, these two NH2 amines which come off, and so you lose one, you lose two nitrogens. So that would be, for instance, two amino acids of the most of the amino acids, like two glutamic acids, two alanines, something like that. You'll lose two nitrogens, one from each, or one glutamine, because remember, glutamine has got two amine groups on it. One that's the R group, hence the gut amine, because it's a primary amide, but also one from the actual amine group of the amino acid, the alpha carbon, central alpha carbon of the amino acid. So there's a few steps that we're going to go through. The first step is, it depends on the textbook whether or not it's considered to have 1 step or 5. There's kind of a pre-step up here at the top. I personally consider that step 1, but some people might consider it as kind of a step 0, like a pre-step or a step 0. And then there's the step 2, 3, 4, and 5. And so I consider it these to be the steps. Some people might consider it as 1, 2, 3, 4, and this step up here is like a pre-step, which I just call A. It depends on the textbook. It depends on how it's presented. But I consider this step 1, the carbamoyl phosphate synthase. I consider that to be kind of step 1. So with that in mind, there are the steps. There's step 1, carbamoyl phosphate, and step 1 is up here, where ammonia and ammonia and bicarbonate come together to make carbamoyl phosphate, and that's kind of step 1 or the pre-step, depending on the textbook. And then the first step of the cycle itself is that carbamoyl phosphate that you made combines with ornithine to make citrulline. The next, so that will be this step here. The next step is that citrulline comes together with aspartate to form agenosuccinate, and that is this step here. And you notice that it requires, like I said, it requires aspartate. And I've colored the aspartate yellow. And I've colored the aspartate yellow because that's where it cross-links and intersects with the citric acid cycle. And that combines with citrulline to produce agenosuccinate. And that agenosuccinate is then cleaved. So rather than condensing 2 things together, you cleave it into that one thing, into 2 things, fumarate and arginine. Arginine as in literally the amino acid arginine, and fumarate as in the TCA cycle intermediate. And so you see that this one's colored in yellow. Well, this is again because that's where it intersects with the citric acid cycle, the TCA cycle. And aspartate is where it comes out of the TCA cycle. And you may be thinking, wait a minute, aspartate isn't in the TCA cycle. We'll get onto that in a moment. But aspartate in a roundabout way comes from the TCA cycle. But then you've taken something out of the TCA cycle, so you need to put back in. And fumarate is what goes back in. Again, not directly. It will actually go to malate and then in as malate, but that will get back into. So this is at the top, it's a cataplerotic reaction, which means to take out of the TCA cycle. So aspartate will come out of the TCA cycle as oxaloacetate, which I'll cover in a little bit. And then fumarate is it goes back into the TCA cycle as an anaplerotic reaction. And like I say, it's also indirectly in that it goes in, it's converted into malate and then goes in as malic acid, but nonetheless, it's going back into the TCA cycle. Anyway, sorry, like I said, you get some arginine. The arginine is then cleaved again to give urea, and the urea is the thing that you're excreting. So it links in with the top. That nitrogen that went in as ammonia comes out as urea at the end, and then that gives you ornithine. And if you remember, ornithine is what you start with. So step 3, when the agenosuccinate is cleaved to fumarate and arginine, that is here. And then the arginine is cleaved again to give you ornithine here and urea here. And there's the arginine. And then that gives you the ornithine, which can come around again. And another way of showing it, if I go through those steps again. So you have the first step, so it's numbered here with the first step. Like I said, it depends on the diagram. In this diagram, it was 1 This is kind of 2A and B, the way they put it, 2, 3, 4. And then this is just kind of like a pre-step of a carbonyl phosphate. And they've just done it as 4 steps. And so it's written here as the 4 steps. And I've just done an arrow for that pre-step. And that's how this textbook presented it. But another textbook is going to present it as step 1 of the carbonyl phosphate. And then the step 2, 3, and 4 is the urea cycle, whereas before they were 1, 2, 3, 4. So it does get a little bit confusing depending on whether or not people consider this step to be the first step. I kind of consider it the first step because in my mind, although it's not part of the cycle itself, it's that pre-step which is contributing to the cycle. And once the ammonia has reacted with CO2 to produce carbonyl phosphate by the enzyme carbonyl phosphate synthase, you're committing to the cycle. And since you're committing to the cycle, in my mind, that is the beginning of the cycle, even though it's not actually kind of in the circular part of the cycle itself. So in my mind, that's the beginning. But the same thing is that in step 1, you have you start out with that ammonia, which you have derived from an amino acid. You have the ammonia that combines with bicarbonate, CO2 or bicarbonate, comes as bicarbonate to give you carbonyl phosphate. That's that step here. And a reaction catalyzed by carbonyl phosphate synthase. That carbonyl phosphate then reacts with the ornithine from the cycle, utilizing the enzyme ornithine carbonyl transferase to give you citrulline. Citrulline then reacts with aspartate to give you arginosuccinate in the arginosuccinate synthase. So you can kind of see where this is going here, where they say the enzyme arginosuccinate synthase, well, you say what the product is in synthase, so that's saying synthesizes that product. So that's the step 2. Sorry, step 3. Step 4 is that now is getting cleaved and fumarate comes off, which is then going to go into malate, and then that's going to go into the TCA cycle, and then that can go around to give you your oxaloacetate, which is going to give you your aspartic acid, which I'll explain in a moment. But that cleavage of the arginosuccinate to fumarate and arginine, catalyzed by the enzyme arginosuccinate lyase. So again, it's saying that, well, what's the thing? Lyase means to cut, so it cuts that thing. So the enzyme is going to split it into fumarate and arginine. Step 4. Step 5, arginase is going to cleave again, and it's going to cleave the amine groups and synthesize urea. And also it's going to cleave the arginine to give you back your ornithine that you started with. Step 5, catalyzed by arginase. And then you've got your ornithine again. So you can just go around in the cycle again if you're provided with a source of carbonyl phosphate. So I'm going to go through each of these enzymes step by step. So first of all, that enzyme, carbonyl phosphate synthase, catalyzes the formation of carbonyl phosphate from ammonia and CO2, which is going to come across as bicarbonate. And there's the ammonia, there's this CO2 that's coming out as phosphate, as bicarbonate, and it's giving you carbonyl phosphate. And you can see where they've come together. There's the ammonia, and here it is in blue. It's been it's reacted with the with a source of carbon, and that source of carbon is through bicarbonate, literally bicarbonate in our blood, the CO2 that is produced in our body, that we breathe, that we produce in our body through respiration. And remember from the TCA cycle where we produce CO2, or from PDH, pyruvate dehydrogenase, we produce CO2, but those carbons are lost. Well, those carbons are reused and added to the amine to produce this molecule here. And you also need a phosphate as well. And that phosphate comes from ATP. and that ATP is going to be cleaved, releasing energy and activating the molecule to produce carbamoyl phosphate here. Requires 2 ATP, and the carbamoyl phosphate can now enter into the cycle, and this is a mitochondrial reaction. I've got a mechanism on the next slide. So again, the first nitrogen enters from ammonia and adds on to the CO2 to produce this carbamoyl phosphate. The mechanism is that first, the bicarbonate reacts this acidic oxygen here, reacts with a phosphate from ATP. So there's ADP, and then there's the third phosphate that makes it ATP. So it's ADP plus phosphate equals ATP. So this acidic oxygen is going to attack the phosphate here. And remember that the terminal phosphate of ATP is a really good leaving group, because the phosphates on ATP, they're all got a negative charge to them. And so they're all repelling each other. And so they really would be quite happy to lose one or two of them. They're quite happy to pop off. And so this oxygen can attack this phosphate, and this bond can be broken, so the phosphate can come off, and makes a very good leaving group. And thus you just, because the ADP has lost its final phosphate, it is now ADP, it's no longer ATP, and that carboxylic acid is phosphorylated. But see, this is not actually where the phosphate comes from for carbamoyl phosphate. I'll go back a slide. Carbamoyl phosphate, like I said, is made up of the amine. It's made up of the CO from CO2, and it's made up of the phosphate. These 3 parts to it. And this phosphate here, it's not the first. It's not this. It's not this phosphate here at the beginning. Rather, what's happening is that this phosphate is coming on to activate the CO2, to activate the CO2 or bicarbonate, in order to make it so it can then be attacked more readily in the next step by ammonia. Ammonia can come in, same kind of thing again, say a nucleophilic attack, whereby the lone pair of electrons on the ammonia acting as a nucleophile, they're attacking this carbon, because this carbon is going to be delta positive, because it's, look at all these oxygens around it, it's going to be delta positive, so it's going to be really ripe to be attacked by a nucleophile. And what's that phosphate that came on earlier, it primes that carbon, makes it more readily attacked by the ammonia. It primes it, and there's also so it makes it more readily attacked. But there's also a very good leaving group itself, so it will then come and leave, get lost. So the thing is that if you just had the NH3, it can attack this carbon here. It's not susceptible enough to attack. It doesn't become susceptible enough to attack until it's phosphorylated. Now that carbon is susceptible to attack. Now it can be attacked by the ammonia once it becomes phosphorylated. But then the problem is that it's phosphorylated as well, and you've got 5 bonds on your carbon, and you can't have that. So what happens is that at the same time, the bond will be broken with the phosphate, because the phosphate makes a really good leaving group. So it comes off as a leaving group, and you have your ammonia and your CO2 that came from your bicarbonate CO2. But like I was saying, this is not carbamoyl phosphate, this is just carbamate. And then the second part, or the kind of the next part of the catalytic reaction, is that it becomes phosphorylated again, a second time, but this time it stays. And the phosphate comes, and same kind of thing, where you have a phosphate and an ADP. That phosphate will get attacked by the acidic carbon, and then the bond will be broken between the phosphate and the ADP, and you gain carbamoyl phosphate. So you can break it down as it does here with the 3 steps. Step 1 is activation of bicarbonate by ATP. to give the carbonic phosphoric acid anhydride. And then step 2 is now makes that carbon highly activated. And then step 2, the ammonia can come in and attack that carbon. And then at the same time, that phosphate that was added on will then leave. So that was only a temporary phosphorylation that activated the carbon. And then in step 3, and so you've generated a carbamate, and in step 3, that carbamate gets phosphorylated, this time permanently, or not permanently, but more long term, not as part of just this 3-step catalytic reaction to produce the product, carbamoyl phosphate. So the reactants are bicarbonate and ammonia and a phosphate from ATP. Those are the reactants, and the product is carbamoyl phosphate. And then in the middle, there's an intermediate where ATP is hydrolyzed to ADP to temporarily phosphorylate and activate the bicarbonate. So that's step 1 in the mechanism of step 1. Step 2 is now where it gets integrated into the urea cycle, and the carbamoyl phosphate can now react with ornithine and come together to form citrulline. And you can see here, you can see that, well, again, there's the same kind of thing where this phosphate is really, its purpose is just to activate this carbon and act as a really good leaving group so that it can react with, so the carbamoyl phosphate can react with ornithine. I'm not showing the mechanism for this one, but just to make it clear that the purpose of being phosphorylated is to activate the carbon and activate it while being a good leaving group so that it can react with ornithine to produce citrulline. And you can see here that this is that activated carbamate here. This is the carbamate that was activated by the phosphate, and it's been attacked by the ornithine. And here's this will react with this, and then here is the ornithine. So this is the ornithine down here, and this is the carbamate, what used to be the carbamate from the last step here, the carbamate. Let me erase all of that. Make that. There's the carbamate. Says what used to be the carbamate or carbamoyl phosphate, and there's the bit that used to be the ornithine, because this nitrogen here can come in and attack this activated carbon, and that phosphate can come off really nicely as a nice leaving group. So that's step 2. And of course, it comes off, that phosphate comes off as PI, inorganic phosphate. Step 3. So that was all up until then. That's all happened in the mitochondria. Step 3, the citrulline is going to exit the mitochondria, and there's going to be a 2-step reaction where, first of all, the citrulline is going to react with ATP to form an AMP intermediate temporarily by arginosuccinate synthase. And then it's going to, that part is going to be cleaved off. And as it's cleaved off, it's going to be attacked by aspartate. Ooh. It's making the whole point of being phosphorylated. In this case, it's phosphorylated by AMP rather than just a phosphate on its own. But the whole reason, again, phosphorylated is to, again, become attacked by another group, while also being a good leaving group. And so step 2A is to become phosphorylated by AMP to produce this AMP citral AMP intermediate, and then get attacked by aspartate. The AMP is going to come off because it's a good leaving group, and then generate this arginosuccinate. So again, it requires ATP because it's getting phosphorylated by AMP, and 2PI will come off as pyrophosphate. And the mechanism is shown in this slide here, where, like I was saying again, you have the citrulline, it's kind of weird with this ATP and it phosphorylates things. Sometimes when ATP phosphorylates things, it will be the terminal phosphate that attaches. Sometimes it will be the terminal 2 phosphates that attach. And sometimes it will be the AMP part of it that's attached. And these two phosphates, terminal phosphates, will come off as pyrophosphate, PPI. But again, the same thing where these react. The citrulline attacks that first phosphate, 1, 2, 3, attacks that first phosphate of ATP. Phosphates 2 and 3 come off as inorganic phosphate. And now you have this citral AMP molecule here, where you can see, let me change color, where you can see this here is this here, and then that primes that carbon to make it ready to be attacked by aspartate, aspartic acid. In this case, it's the ionized salt, so it's going to be aspartate. And these lone pair of electrons can again come along, attack this activated carbon. That phosphorylation from AMP makes a really nice leaving group, so it can come off. So this is that 1A step that's listed in the cycle on the previous slide. That sorry, not 1A. I mean it's part A and part 1 of this reaction, but it's 2A, I guess, in this. So here it is here. So that's part A, and then the part B is that that activated carbon can be attacked by the aspartate. That AMP group can leave here, and now you have the aspartate here, and you have the and then you have the arginosuccinate products produced from the aspartate and the citrulline, producing arginosuccinate. So if you want to track the nitrogens as well, you can follow the nitrogens all the way through from the start, where we started out with the nitrogen attached to this. The nitrogen is the blue one. The carbon is from the CO2 is the yellow one. So the ammonia is in blue. The CO2 carbon is in yellow. We can track it through in citrulline. And then if we come through to the citrulline, here is this one here. This is what was the ammonia nitrogen. And this here is the CO from CO2. This all comes from this part here. And in the citrulline, we can now see that this here is the ammonia nitrogen. This carbon here is the carbon from CO2. And this nitrogen here is the ammonia that came from aspartic acid, the new one. That's just the nitrogen that came from aspartic acid, it's the new one. And finally, I'll change color. This nitrogen here is one of the new ones that came from in the citrulline, that came from the ornithine. It's this one here. This nitrogen here came from the last step with the ornithine and the carbonyl phosphate to form the citrulline. And so that nitrogen there is that nitrogen there from ornithine. And I guess if I change color again, these are this nitrogen here is the other nitrogen from ornithine is this one here. So this really is ornithine. And then this here is citrulline. And then the whole thing together is arginosuccinate. And you can track the ammonia and the CO2 from even further back. So you can track all of the carbons that have come from these molecules. You can track them through. And then in the next step, we've made that arginosuccinate here. And like I said, this one is a little simpler. We're just going to cleave it. Not much of the mechanism is going to be shown here, but it's going to be cleaved. Here, you get the arginosuccinate. It's going to be cleaved, and that gives you the fumarate that comes off. But also here, you're going to get the arginine. So this here is the amino acid arginine, one and the same amino acid arginine, as in the proteogenic amino acid. So we produce arginine. And the thing with arginine is that it has these 3 amine groups in it. It's that very nitrogen-heavy basic amino acid. And we can now start to kind of cleave off some of these nitrogens and turn them into urea. So in the at the end, this is where are we up to? So we've made the arginosuccinate is cleaved to fumarate and arginine, and then with the enzyme arginosuccinase, and then isn't it? Sorry, it's arginosuccinate lyase. Yeah, arginosuccinate lyase. Sorry, that was throwing me off. It's arginosuccinase. It's arginosuccinate lyase. I was being thrown off because I was gaslighting myself that something isn't quite right here. Arginosuccinate lyase is going to lyase, so split into fumarate and arginine. And now you've got that no mechanism really shown for this one. And then that arginine is going to be split again by arginase. And this one actually is called arginase, just called arginase. And you're going to see that this terminal group here of the R group is going to be cleaved off as in a hydrolysis reaction requiring water in order to give you urea. And then that urea comes off as the product. But if you cleave that part off, then that gives you ornithine, which is the beginning of the cycle again. And then that ornithine can go back into the mitochondria to redo another cycle. So you can kind of think of ornithine as being arginine, but without the terminal urea part of it, because the urea part of it has come off. Or you can think of ornithine as being just like arginine, but with another group added onto the end. So ornithine is just arginine without urea on the end. And we can track these carbons as well because this here is the carbon from CO2, and then we have the nitrogens that have come from the previous steps, that have come from the ammonia at the beginning, and also have come from the aspartate as well. We can track them. So we have the two. We have the ammonia. Where have I gone? You have the ammonia nitrogen, and you have the aspartate nitrogen attached on as well. So you've lost your 2 nitrogen groups, and rather than having them around as ammonia, they are now added to that urea, which is less toxic and more easily excreted, whereas ammonia is harder to excrete and more toxic. So we've taken those nitrogens off, turned them into urea, utilizing CO2, and produced the urea, which can be excreted, and reproduce the ornithine so we can go around with the cycle again. Now, like I said, this is linked to the TCA cycle. And you can see here that if we start out with our carbonyl phosphate, add its ornithine to give you citrulline. Citrulline exits the mitochondria and enters into the cytoplasm. And then that citrulline will come together with aspartate to produce the arginosuccinate. Where did that aspartate come from? Well, it came from oxaloacetate in the citric acid cycle. And that oxaloacetate is converted into aspartate. And you may be wondering, well, what's the link there? And the link there is that if we look at oxaloacetate, and this is a keto acid, right? Remember, a keto acid is one that has got this group here. This is a keto acid. So it's a keto acid group. So oxaloacetate is a keto acid. Now, if we were to take this acidic group here and we were to turn it into an amine instead, then what we have is oxaloacetate is turned into aspartate. So if we take the ketone group of oxaloacetate, turn it to an amine instead, then it turns into aspartate. They are related to each other. Aspartate is just an aminated oxaloacetate. And oxaloacetate is just made by the oxidative amination of aspartic acid. And this is another aminotransferase that we talked about last time, where the amine group from glutamate is transferred onto oxaloacetate, and that generates aspartate. And in doing so, glutamate has lost an amine group and instead turned into a ketone group, and that ketone group is alpha-ketoglutarate. So oxaloacetate into aspartate, and at the same time glutamate into alpha-ketoglutarate. That aspartate then exits the mitochondrial matrix into the cytoplasm, where it reacts with the citrulline to produce the arginosuccinate that can go on to the next steps. But as part of that, remember that the arginosuccinate also gets cleaved, and when it gets cleaved, it's cleaved to fumarate. That was that step that we talked about here. Arginosuccinate is cleaved. Remember, that was that lyase where the slide is wrong. It says arginosuccinase, but it's arginosuccinate lyase. At lyase, it cuts off, and you get a fumarate and arginine. But that arginine, that fumarate comes off, and that fumarate can be converted into malate, and then that malate can enter into the mitochondria, just like the malate shuttle that we talked about a few lectures ago when we talked about gluconeogenesis. Remember that malates can go in and out of the mitochondria, and that malate can then go into the mitochondria, and then can enter into the TCA cycle as malate. It can go in as fumarate because there is not a transporter for fumarate. There is not a transporter for fumarate. It needs to be converted into malate first. And that's really probably just to do with keeping metabolites in the mitochondria. They want to selectively be able to put malate in and out, but not fumarate. So there is a transporter for malate. There is not a transporter for fumarate. And yeah, there has to be a transporter because mitochondria are membrane bound. And so you have that lipid membrane, and the lipid membrane is not going to be permeable by something, many molecules. So, and then the rest of the urea cycle can just go around. So the TCA cycle is linked to the urea cycle in that when you get the citrulline, needs a source of aspartate, and that aspartate comes from oxaloacetate in the TCA cycle that's been exported out into the cytoplasm. But don't worry, because in the very next step, the arginosuccinate is cleaved to fumarate. That fumarate is converted into malate, and that malate is imported back into the mitochondria again, and that malate can re-enter. So it's kind of like these two cycles. These two cycles which intersect, where you have one cycle here and another cycle here, where they come together and then split apart. They kind of come together and then they split apart again. And all that's happened is that you've just transferred carbons between them because you need some of those carbons in order to complete the urea cycle. So that really covers the 4 or 5, depending on how you look at it, steps of the urea cycle. Just to kind of finish it up and frame it, just a couple of facts. The activity is going to be highly regulated. It's going to be regulated at the point of making the actual enzymes. So there's 5 enzymes that are involved. The 4 of the circular part of the cycle and the 1 of the pre-step. And so you can control whether or not you make these enzymes and to control the physical levels of those enzymes, because if there's low levels, then the cycle will be lower. There's high levels, the cycle will be higher. And so if the human body or the cell, the animal cell, if the cell wants to control the rate of urea synthesis, then it can just control how much of the enzymes are present, because the enzymes are required to catalyze the reaction. Also, allosteric regulation, carbonyl phosphate synthase, which is that step 1 or that pre-step, depending on how you look at it, where the CO2 and the ammonia come together to make the carbonyl phosphate, utilizing 2 ATP, one of them for activation, one of them for temporary activation, one of them for longer term activation as the phosphate part. And also allosteric regulation of that enzyme. Remember, allosteric regulation, allosteric means outside of the active site, so it's not a competitive inhibitor, but it's something that perhaps binds to the enzyme, changes its shape to make it so that the reaction isn't catalyzed anymore, or catalyzed at the same rate. And that could be because the active site has often because the active site has changed and changed shape, so that it will no longer react. And the nature of that depends on the nature of the allosteric regulator. But a negative allosteric regulator is going to make it so that the reaction will go slower. A positive allosteric regulator will make the rate go faster. By changing something, the way that the enzyme reacts with its substrates, either making the substrates not fit as well, or slowing the reaction in some way or other, depending on the nature of the regulator. This reaction happens in particular, or really it happens in the liver, and it requires this ALT and AST, which are liver enzymes, because you need the AST, remember, in order to generate aspartate from oxaloacetate to push forwards the reaction. But also, as well, you're going to need this alanine as part of the Cahill cycle, which remember from before, Cahill cycle is how the muscles get rid of their excess nitrogen and transport it to the liver, in that ammonia is put onto the muscles have got pyruvate. That pyruvate is transaminated to alanine in the muscles. The alanine is transported to the liver and then converted back into pyruvate in the liver. And then the ammonia is then indirectly comes off and enters into the urea cycle. But in doing so, you break down the alanine in the liver to pyruvate. That pyruvate can then be turned into glucose and sent back to the muscles. I talked about that in the last lecture. That's called the Cahill cycle. But so you have high levels of ALT for the Cahill cycle. You have high levels of AST to make aspartate. So in the liver, there's high levels of ALT and AST. They need them. So if you have a lot of these in your blood, that's an indicator that perhaps some of your liver cells have died, and it can be an indicator of liver health. And so if you go for a blood test with your doctor, they might look for ALT and AST levels, and a high level might indicate liver damage from often because of diets. You have like a fatty liver, fibrosis because of some kind of liver disease, such as hepatitis or the hepatic viruses, with diets with fat, virus, or alcohol, which will cause scarring and cirrhosis of the liver. Also due to muscle degradation. Bodybuilders might have high levels of ALT in particular, less so AST. They can have high levels of AST, also high levels of ALT, because bodybuilders are going to be working out a lot. They get muscle damage, which is part of muscle growth and hypertrophy, and so they might have high levels of ALT in the blood. And often that could be the difference between someone who's working out a lot or someone who has liver damage. If the ALT is higher than AST, then it indicates that maybe the muscle is breaking down rather than the liver is breaking down. Although that is not medical advice, that is just a kind of general trend that I'm telling you about. That covers the urea cycle. And that's everything to do with the urea cycle. So I'm now going to go into the next thing, switching up gears, and we've taken off the amine groups and got rid of those amine groups in the urea cycle from amino acids. But what are we left with? We're left with the carbon skeletons. And those carbon skeletons can then be utilized for energy, really mainly by entering into the TCA cycle. So this links in with what I was my key concepts. And key concepts number 5 from the beginning. Each different amino acid has a different catabolic fate. So really that amino acids are broken down, and we're left with carbon skeletons, but those carbon skeletons are going to have different structures because of the different R groups. And those carbon skeletons, including the R group, amino acids minus the nitrogen, those are going to be utilized, enter into the TCA cycle. So this has come off as urea, and you've now got this left over, which is going to go into the TCA cycle. Because the R groups are different, it's going to enter into the TCA cycle in different ways. Most of them can actually contribute to gluconeogenesis because they contribute enough carbons that they contribute enough that it's enough carbons that it can be turned into glucose. But leucine and lysine cannot. They can only contribute to ketogenesis because they don't contribute enough carbons. We'll talk about what that means in a moment. But amino acid catabolism accounts for about 10 to 15% of human energy production, with the rest being made up, of course, by glucose metabolism and the sugars in general, sugar metabolism, especially glucose metabolism, and fatty acid oxidation, like I talked about a couple of lectures ago. But about 10 to 15% of energy production comes from breaking down those carbon skeletons and put them into the TCA cycle. There's 20 different catabolic pathways, because with this amino acid catabolism, we're talking about the 20 proteogenic amino acids. Of course, there's other amino acids, but mainly we're talking about the 20 proteogenic amino acids, those amino acids that are used to make protein. It's kind of 20 plus 1, but the plus 1 is cysteine, which is really just cysteine. So we'll just consider it 20 for this. But there's going to be 20 amino acids, so there's going to be 20 ways of breaking them down. But of course, catabolic processes converge. And rather than having 20 different pathways to make something that goes into the TCA cycle, instead, it's much more efficient if we converge those pathways into similar pathways, which then go into the TCA cycle. Because then rather than having 1, 2, 3, 4, 5, 6 different enzymes for each different amino acid, which is going to produce, and I'm just saying 6, it could be, 5, 6, 7, 8, 9, 10, whatever. Let's say that there's 5, make it nice and easy. Maybe it says there's 5 different enzymes, 5 different steps. Imagine there were 5 different steps to break down each of the 20 different amino acids. Well, that's going to be 120 different enzymes in order to break down all the amino acids. That's going to be kind of rough. Instead, it'd be much easier if we just have enzyme 1, 2, 3 for each different amino acid, and then enzyme 3 just turns it into a common product, and then you just have maybe another 1 and 2 to turn it into a TCA cycle intermediate. And that way, you require a lot fewer enzymes, by converging it into one common product, you require a lot fewer enzymes. And then once you have that one common product, you can just use one or two enzymes to turn it into a TCA cycle intermediate. So a lot less is required. It would be, for instance, if there's 1, 2, 3 for each different amino acid, that means that you only require 60 different enzymes in order to turn it into that one common product, and then plus 2, so 62, in order to turn it into a TCA cycle intermediate. So it'll be, for example, 62 different enzymes required instead of 120 different enzymes, half as many enzymes. Actually makes things a lot simpler by converging into a common product and then turning that common product into the thing that you actually want. In reality, it's even less enzymes than that because, for instance, some amino acids can turn into other amino acids and then share the same pathway. So it makes it even simpler still. We're going to get onto that in a minute, though. But of those 20 different amino acids, they mainly converge to form 6 major different things: pyruvate, acetyl-CoA, alpha-ketoglutarate, succinate-CoA, fumarate, and oxaloacetate. We've already talked about a few of these because we've already covered that alanine turns into pyruvate. We've already covered that aspartate turns into oxaloacetate. So a lot of this we've already done. Glutamate into alpha-ketoglutarate. And of course, you can go from glutamine into alpha-ketoglutarate. And these are all TCA. And pyruvate goes into acetyl-CoA or into oxaloacetate to enter into the TCA. So they're all just turning into things that can enter into the TCA cycle. And this is summarized here. So, for instance, I'm going to cover all of these in a little bit more detail. Arginine, glutamine, histidine, proline can all be converted into glutamate and then glutamate into alpha-ketoglutarate. So rather than having a separate pathway for arginine, separate pathway for glutamine, separate pathway for histidine, separate pathway for proline, that may require 2, 3, 4, 5, 6 different steps until you get to a TCA cycle intermediate. Instead, if you just convert them into glutamate, and then you can just do one step to turn glutamate into alpha-ketoglutarate with that various different transaminases or GDH, glutamate dehydrogenase, whichever, there's a few different ways. Same thing with isoleucine, methionine, threonine, valine are converted into succinyl-CoA. Phenylalanine and tyrosine into fumarate. Asparagine and aspartate into oxaloacetate. Alanine, cysteine, glycine, serine, threonine, tryptophan are converted into pyruvate. And then with pyruvate, you can go 2 ways. You can either pyruvate dehydrogenase into acetyl-CoA, or pyruvate into pyruvate carboxylase into oxaloacetate. And remember, that was to do with anaplerosis and topping up the 4-carbon contribution to the citric acid cycle. So you can you get the 3 carbons of pyruvate, and you can either turn it into 4 carbons utilizing a CO2 and contribute 4 carbons to oxaloacetate, or pyruvate to acetyl-CoA, which is 2 carbons and 1 carbon coming off as CO2 as part of the pyruvate dehydrogenase reaction. And then those 2 carbons can enter into TCA cycle by combining with oxaloacetate to make citrate. And then you can have, or you can make acetyl-CoA from isoleucine, leucine, threonine, tryptophan, or you can make acetyl-CoA, which is a keto acid, a ketone body, which I mentioned in the last lecture of 2 lectures ago, the last lecture of fatty acid catabolism. We have an excess of acetyl-CoA, and you can turn it into acetyl-CoA, but you can also go back again. You can go back and forth, and this is a ketone body which can be transported around the body. And that acetyl-CoA can be made from leucine, lysine, phenylalanine, tryptophan, and tyrosine. What you may notice is, wait a minute, some of these appear twice. And the answer is, yes, they do. And that's because those carbon skeletons can often be split into 2 things. For instance, let's find one. Let's find a nice one. So here we go. Oh, tyrosine. Tyrosine can make acetoacetyl CoA, so it's for acetyl CoA to enter in, but also tyrosine can also make fumarate. And the answer is that the R group is split into two, and part of the R group makes fumarate, and part of the R group makes acetoacetyl CoA. So some of them appear twice. The other thing is you'll notice that some of the boxes they've got this kind of pinky salmon color, and some of them got the blue color. The ones that are pinky salmon color are ones that are considered glucogenic. And they are glucogenic because they contribute 3 or more carbons into the TCA cycle. And the key rule is that if it can contribute 3 or more carbons into the TCA cycle, then it is considered a glucogenic amino acid because it could be converted into glucose. Because you can convert it into various different things, and let's tidy this up a little bit. You convert it into various different things which have more than 2 carbons, and then they can be converted into glucose by gluconeogenesis, like we covered a few lectures ago. So these amino acids can make their way into glucose. If we were to label some of these amino acids with isotopic labels, then we'd be able to see those isotopic labels in some glucose that the liver has produced by gluconeogenesis. However, these amino acids in the blue are either ketogenic only in the case of leucine and lysine, or ketogenic and or glucogenic, depending on how they're metabolized and if they get split into 2 or not, to produce 2-carbon acetyl CoA. The 2-carbon acetyl CoA can never be turned into glucose. Other organisms can do it, but animals, mammals cannot. Plants in particular are the ones that can turn acetyl CoA, and fungi acetyl CoA into glucose, but we cannot. And so it can only turn into acetyl CoA. And so we call those ketogenic because we have the acetyl CoA, we can turn it into acetoacetyl CoA ketone body. And that ketone body can be metabolized by various tissues into acetyl CoA, so it can be utilized to make energy, but it can never be made into glucose. You can never go backwards from that 2-carbon into glucose in mammalian in the human body. So leucine and lysine are the only ketogenic. Everything else is either purely glucogenic or glucogenic and ketogenic because the R group gets split. That's kind of summarized here on this slide. Ketogenic amino acids can yield ketone bodies in the liver. These ones can yield ketone bodies in the liver, but remember that leucine and lysine are ketogenic only, whereas glucogenic amino acids can be converted into glucose and also glycogen from the glucose, and all of them can do that actually, except for lysine and leucine. Remember, this is for the 20 proteogenic amino acids as well. There's other amino acids, like for instance, some citrulline. Citrulline is an amino acid, it's an amino and acid group in a roundabout way. And ornithine is a more classic one that more so resembles an amino acid. Ornithine really does resemble an amino acid because it's just arginine, but without the kind of CO and carbon and the 2 amine groups attached to it. It's just been kind of had its end chopped off. But anyway, of the 20, we're talking about, the point is we're talking about the 20 proteogenic amino acids. So I'm going to go through these a little bit, but not in a ton of detail, because there's just a lot here, and there's a lot that would have to be memorized. And for my biochemistry 2, I don't require you to memorize this, but I would like you to have the conceptual knowledge that something is going on here, and that the carbon skeletons can be broken down into TCA cycle intermediates, and that many of these pathways overlap for efficiency. Of course, with many of the metabolic enzymes, you're going to require a lot of cofactors, in particular things like biotin, tetrahydrofolate, and adenosylmethionine. Sorry. And many of these, of course, are vitamins. or things that you can take in health food supplements. Tetrahydrofolate, I believe, is it vitamin B3, derived from folic acid, which I believe is vitamin B. No, vitamin B3 is niacin. Maybe it's 4 or 5. Maybe someone can remind me. That one's lost my memory. But biotin is a common, is a cofactor and a common health food product. And this SAM, it's often abbreviated as SAM, SAM. Adenosylmethionine. There we go. S-adenosylmethionine, SAM. I always just call it SAM. There's very few times in my life where I've had to say it as the full name. SAM is also getting more popularity as a health food product that many people take. And low levels of SAM are linked to various different diseases. But anyway, the same thing, you know, B vitamins, they say, are for energy because they're cofactors in making energy and breaking down amino acids into TCA cycle intermediates. So I'm going to go through in general terms and summarize the breakdown of these 20 proteogenic amino acids and the pathways for that. So first of all, like I said, 6 amino acids are degraded to pyruvate: ALA, tryptophan, cis, ser, gly, threonine, converted in whole or in part to pyruvate. That means that maybe part of them will be cleaved off to make other things, or maybe the whole thing will turn into pyruvate. And of course, that pyruvate can either go one of 2 ways. It can go by PDH, producing CO2, produce acetyl-CoA to go into the TCA cycle, or with pyruvate carboxylase, PC, requiring bicarbonate, which I'll just put CO2. I should be better. That is going to the PC into oxaloacetate, pyruvate into oxaloacetate, but both of which enter into the TCA cycle. So you have that 3-carbon into a 4-carbon or 2-carbon. Oxaloacetate for gluconeogenesis, acetyl-CoA for either for further oxidation in the TCA cycle or into ketogenesis. The general pathway for those 6 amino acids, like I say, is shared. So, for instance, if we go from threonine down into pyruvate, there are 1, 2, 3, 4 different enzymes to turn threonine into pyruvate. And as part of that, threonine also produces acetyl-CoA as part of a lyase reaction, where it's going to, the 2 carbons going to come off as acetyl-CoA, leaving this group here. So this will come off as acetyl-CoA, and this is left over from threonine as part of the step 1, step 2 of threonine degradation. So you've produced acetyl-CoA. So threonine is both ketogenic and glucogenic. It produces both acetyl-CoA, but also eventually pyruvate. But then when we come down into step 3, it then reacts and will produce serine. And so now we have serine at step 3. But also that requires glycine. So glycine will join up with the kind of the metabolite from threonine in order to make serine. So remember, serine is a non-essential amino acid. It's non-essential, meaning we don't have to get it from the diet. It's essential for life, but we don't need it from diet. That's because you can make serine. You can make it from threonine and glycine. And then once you've kind of made that serine, it's only 1 step to turn serine into pyruvate with serine dehydratase. So what you've done is that you've converted threonine into serine, you've converted glycine into serine, and then just 1 step to convert serine into pyruvate. So rather than having multiple steps to go from threonine into pyruvate, you convert it into one common thing, serine, and then that one common thing, serine, is converted into pyruvate. Similarly, tryptophan is converted into alanine, and then alanine just one step into pyruvate. Similarly, cysteine, 2 steps into pyruvate. So there's these multiple amino acids are converted into pyruvate, and many of them overlap. Alanine, cysteine, and serine are converted directly into pyruvate. Tryptophan is converted into alanine, and then alanine into pyruvate. And threonine and glycine are converted into serine, and then serine into pyruvate. So they all kind of overlap and converge in order to make this common metabolite that can enter into the TCA cycle. As you can see, highlighted there in the yellow is alanine, cysteine, and serine converted directly into the pyruvate, but also tryptophan, threonine, and glycine converted into their respective metabolites, which are in turn converted into pyruvate. And this allows for the same paths to be used in order to make things more efficient. So that covers those 6 amino acids that convert into pyruvate. As well as that, several amino acids are converted into acetyl-CoA. 2 of them, isoleucine, leucine and lysine, are converted into acetoacetyl-CoA and then into acetyl-CoA only, whereas phenylalanine, tyrosine, and tryptophan, part of them are glucagonic, but part of them into acetyl-CoA, whereas isoleucine, leucine, and threonine, tryptophan, form acetyl-CoA directly, not via acetoacetyl-CoA, that ketone body. You'll notice that leucine appears twice. That is because leucine makes one acetoacetyl-CoA and one acetyl-CoA. So it ends up making 1 plus 2 and then 3 acetyl-CoA. It makes 3 acetyl-CoA. 2 in acetoacetyl-CoA and 1 as acetyl-CoA on its own. It produces both. Lysine just produces the acetoacetyl-CoA on its own. And this is kind of demonstrated here, where you see that tryptophan and lysine are converted into acetoacetyl-CoA, but the tryptophan is split off. Some of it is converted into alanine, and some of it is converted down into acetoacetyl-CoA, and that alanine, of course, can then go into pyruvate, which is the one that we talked about here, tryptophan to alanine. So part of it goes into alanine, and part of it goes into a ketone body. This is that ketone body here. On the other hand, phenylalanine converted into tyrosine. Tyrosine, part of it comes off as fumarate, but part of it combines with acetoacetate to produce acetoacetyl-CoA. Leucine, part of it comes off acetoacetate, and then part of it comes off for acetyl-CoA. Not acetoacetyl-CoA, acetyl-CoA. Isoleucine, it makes acetyl-CoA, but also splits off, and part of it makes acetyl-CoA, part of it makes this propenyl CoA, which is then converted into succinate CoA. But that propenyl CoA can also be used for fatty acid metabolism, which I haven't spoken about here, but that's the beginning of that can be used for the beginning of making fatty acids. Fatty acid synthesis. So leucine, lysine, phenylalanine, tyrosine, and tryptophan directly yield yield acetyl-CoA via acetyl-acetyl-CoA. Isoleucine, leucine threonine, and tryptophan form acetyl-CoA directly. The red carbons are destined for ketones. Here, red carbons for ketones, and blue end up in the citric acid cycle. For instance, you see here these four come off into fumarate. Most have a mixture, right? Most have a mixture. Some of it for citric acid cycle, and then some of it, on the other hand, for ketones. But two don't, and again, that is the lysine and the leucine. Okay. So we're almost done, actually. I'm just going to talk a little bit about, I just wanted to briefly mention in passing, this is actually just really just flavor, tryptophan breakdown. This is just saying that tryptophan, it's got these two rings, and these two rings are kind of complicated. And so there's a bunch of things that we can do with these two rings. We can utilize these two rings. You can use it, for instance, to make neurotransmitters. It's not just serotonin. You can also make a variety of them. They call them monoamine neurotransmitters. Serotonin is a typical one, but also things like dopamine. Dopamine is related to these as well, and the one that helps you melatonin as well. One helps you sleep. So it's all kind of related to these, and a lot of these neurotransmitters can be made from tryptophan. But also tryptophan can be used to make niacin, which is vitamin B3. Also, plants might utilize it as well. This is largely just flavor, just to say that you don't have to break down tryptophan to make for the carbons to enter into the TCA cycle. You can also utilize it to make other useful things, such as vitamin B, niacin, such as neurotransmitters, such as serotonin and melatonin. I remember I said, remember you may have been heard that drink milk or eat a turkey sandwich before going to sleep to help you sleep better. And then you may have heard it been mentioned because of the tryptophan that's present. That's because the tryptophan makes melatonin, and the melatonin will make you sleep. That's the idea behind it. This slide is largely just flavor. I just wanted to let you know that it's not just for the citric acid cycle, but also make other useful things. The pathway, like I said on the previous slide, is complex. Pathway, if you want to break it down, catabolism, it's most complex with tryptophan. And where it's that most complex, that means that you've got several areas where there can be errors. And this also comes up with phenylalanine and tyrosine, which are also got these cyclic rings. You can see that there's a lot of enzymes to break these down, highlighted there in the blue. Where these enzymes to break them down can go wrong, then that will cause some kind of disease, a metabolic disorder. And these come up quite a lot in medicine, where they're referred to in medicine as inborn errors in metabolism. Where's my pen? Inborn errors in metabolism. And these are often things which, it says here, genetic defects. I prefer to just call them polymorphisms, where a person has a polymorphism in their genetic code, which means that they produce a different version of the enzyme. This enzyme may be inactive or lower activity, and this can cause a metabolic disorder in the infant. That has not caused infant death, but so the infant is viable when it's been born, but may have a metabolic disorder, and so things need to be monitored, and then the treatment may be necessary or diet modifications in order for the best health. And most common one is, you guys may have heard of PKU, phenyl ketonuria, and this one you may have seen on soda bottles. It says contains a significant source of phenylalanine, or does not contain a significant source of phenylalanine. That's related to PKU, And that's related to the fact that some people have this disease because they have lower levels of phenylalanine hydroxylase enzyme, which means that they can't break down their phenylalanine. And so levels of it will build up, and that will cause disease. But there's other diseases as well, where in these inborn errors in metabolism that come up a lot in medicine. If you go to med school, this will be a major part of it, where there are metabolic disorders caused by deficiencies in activity or levels of certain enzymes, usually because of a genetic polymorphism, a change in the genetic code. That means that you can't break down certain things well. This is especially prevalent in complicated metabolic pathways of amino acids, especially in complicated ones like phenylalanine, where it may not cause death, but it may cause disease. And like I said, I gave the example of PKU, which is that's phenylalanine hydroxylase, lower activity or lower levels, increased levels of phenylalanine, which can cause, the main thing is it causes a problem in brain development. And so children won't get proper brain development. So if they have this low levels of this enzyme, phenylalanine will increase, will cause problems in brain development, and so should be avoided, but it's easily treated with dietary intervention, which means often these kids can't drink sodas because the sodas contain phenylalanine, which is usually present actually as the sweetener. Aspartame, the sweetener aspartame is made out of phenylalanine, and it will break down into phenylalanine and raise the levels in the child's blood. And if they can't break that down, that isn't good and will cause problems with their brain. So they have to eat special diets that have got low levels of this enzyme. But as long as they do that, they're going to be fine. Again, it's not medical advice, just saying. Another one is this alkoptonuria, which is caused by this deficiencies in the lower levels of activity or raw levels of this enzyme, which means that this metabolite, which is here, this list comes down and then back up again. So this and this are the same thing. And low levels of this metabolite in tyrosine and phenylalanine metabolism. There's the enzyme here. Low levels of this enzyme or low activity of it will cause buildup of this metabolite, homogentisate, and let's see, top right of pathway, 2 slides ago, and it produces high levels of this metabolite. And this metabolite is not super toxic, but not ideal. It will just accumulate in the body if you can't break it down. And these oxygens here can be oxidized. They will be oxidized to the ketone, from the alcohol to the ketone, kind of like between ubiquinone and ubiquinol. And this will be the ol version. What's shown here is the ol, and this will be the one. So these groups can be oxidized. When it's oxidized, that might turn the urine from being well, this person's a bit dehydrated. It should be a little bit more clear than that, but from a pale yellow to a dark color. And so this disease can be diagnosed by dark urine. So that was those aromatic amino acids. Almost done. Next one's just the 5 amino acids that are converted to alpha-ketoglutarate: arginine, glutamate, glutamine, histidine, and proline. And this is generally because things like arginine, histidine, and proline can be converted into glutamate. Glutamine can be converted into glutamate using glutaminase, which I talked about in the last slide. Then once you've got glutamate, it's just one step with glutamate dehydrogenase or one of the various different aminotransferases to take that amine group off and turn it into alpha-ketoglutarate. Remember, glutamate is a common thing that for transporting ammonia in the blood, because ammonia can be, because the glutamate can carry that amine group. It's not super toxic. It will go to the liver, and then GDH, glutamate dehydrogenase, will take off that NH3 as ammonia. That then goes into urea in the urea cycle and generates alpha-ketoglutarate, which enters into the TCA cycle. Remember also as well that arginine is degradation is part of the urea cycle. So when we break down arginine, We can also, we don't have to take it from the oxaloacetate. If we have enough arginine, we can turn it into ornithine, and then that ornithine can be as part of the urea cycle, or we can break it down into glutamate semi-glutaraldehyde, and then turn that into glutamate itself. And then almost done. 4 amino acids are converted into succinyl CoA. These are isoleucine, methionine, threonine, and valine. Methionine, isoleucine, threonine, and valine. And again, the same thing, where some of these pathways overlap. And they particularly overlap at alpha-ketobutyrate and propanyl CoA, which is then converted into succinyl CoA, which of course is a TCA cycle intermediate. So these pathways are overlapping and utilizing the same enzymes and converting into similar things, so that the same things, so that they can for more efficiency. So you don't have to have a separate 10 steps in order to turn isoleucine into succinyl CoA. Just convert it to propanyl. Just convert it to propanyl into propanyl some way, and then that propanyl can be converted into succinyl CoA. I believe that this is the end. The final bit is just that the branched chain amino acids, they are often not broken down in the liver, because the branched chain aminotransferase BCAT is not present in the liver. Instead, so it's instead degraded in the extrahepatic tissues. So a lot of these amino acids, the first step is taken off the amine group, and that amine group goes into the urea cycle. But it's different for the branched chain amino acids because the aminotransferase isn't present. Instead, it's often broken down instead in the muscle, which is the key area, but also the fat, kidney, and the brain, absent in the liver. And so it will just convert it into the branched chain amino acids into the branched chain keto acids. Remember, when you go from an amino acid and you have an aminotransferase, you take off the amino acid, you convert it into a ketone instead. And then that ketone will be broken down in the liver into various different metabolites, but we'll break it down into various different metabolites by different tissues. But this first step in particular is not going to happen in the liver. It happens in the other tissues instead. Something that's and just to mention, what's kind of interesting about this is that so the branched chain amino acids convert the amino acid into the branched chain keto acid, 3 branched chain keto acid using the branched chain aminotransferases. And these keto acids are then broken down further. But what's interesting is that what breaks them down further is that you have this keto dehydrogenase. And the C is complex, keto dehydrogenase complex. And it's a branched chain keto dehydrogenase complex. And the branched chain keto dehydrogenase complex is kind of similar to the pyruvate dehydrogenase, and also the alpha-ketoglutarate dehydrogenase. And it turns out that there are 3 enzymes, 1, 2, 3, that convert pyruvate, alpha-ketoglutarate, and branched chain keto acids into their final product. And enzyme 2 and 3 is shared between all of those 3 different complexes. And it's just that enzyme 1 is different from the 3 different complexes for the different initial substrate of pyruvate, alpha-ketoglutarate, or the branched chain keto acids. But enzyme 2 and 3 are the same in order to make the same kind of CoA derivatives at the end. And it's evolutionary that 3 enzymes are required to convert all of those pyruvate, alpha-ketoglutarate, and branched chain keto acids into their product. There's 3 enzyme complexes are required for them, but evolutionary enzyme complex 2 and 3 are the same. It's only complex 1 that's different. And if you recall from the TCA cycle lectures that we did, and the pre-step for the TCA cycle, the linker step between glycolysis and the TCA cycle, where pyruvate is decarboxylated into pyruvate 3-carbon into the 2-carbon acetyl-CoA and CO2 comes off. That requires that 1, 2, 3 protein complex, and proteins 2 and 3 are exactly the same as 2 and 3 in alpha-ketoglutarate dehydrogenase and branched chain keto acid dehydrogenase as well. And it's only enzyme 1 which makes it specific for pyruvate, enzyme 2 that makes it specific for alpha-ketoglutarate, or enzyme 1 of one, enzyme 1 of the other, which makes it specific for alpha-ketoglutarate or branched chain keto dehydrogenase. So I thought that was kind of interesting as well. Almost done, I promise. Penultimate slide. If you don't have high enough levels of the branched chain keto acid dehydrogenase, then the branched chain keto acids will build up, and high levels of these will build up in the blood, and they'll be spill over and carried into the urine. And so these actually have a maple syrup smell. And so maple syrup urine disease, MSUD, maple syrup urine disease, is characterized by high levels of branched chain keto acids in the blood, because the branched chain dehydrogenase, step 2 in the branched chain amino acid catabolic pathway, is deficient or in some way lower activity or lower levels. And so the keto acids build up in the urine. And this again can cause problems in children in particular, because it's important in brain development, but also give the urine a maple syrup smell, because apparently these smell like maple syrup. But again, can be controlled by the diet. And then finally, asparagine and aspartate are degraded to oxaloacetate. And remember, we've covered that aspartic acid is into oxaloacetate, and asparagine into aspartate. So you can just use this same common aspartate aminotransferase to generate oxaloacetate. And that covers everything I wanted to talk about amino acid oxidation. That was a lot to take in, and I went a bit fast at the end, but I hope this was useful and you learned a lot from it.